Today we're going to be talking about the cell process called mitosis. Remember last time when we were talking about the cell cycle, and we said that that can be defined as all of the events that lead up to the formation of two daughter cells from one parent cell. We said that everything inside the parent cell has to be replicated so that each daughter cell gets a full complement of all of the components of the parent cell. The cytosol, the fluid in the cytoplasm, has to be copied. The cytoskeleton has to be copied. All of the organelles have to be copied. Any dissolved proteins have to be copied. Everything. Everything that's inside that cell that makes it what it is. And of course, the other thing that has to be copied is the DNA. Each chromosome has to be replicated. Then the nucleus has to divide into two. And then the cytoplasm can divide into two, producing two daughter cells. Now, we said that the cell cycle has four distinct stages and a fifth stage where the cell cycle can arrest. The first three stages, G1, S, and G2, are often grouped together and called interphase. And the last stage is called M. And that M can stand for either mitosis, if we're talking about a somatic cell that's dividing, like a body cell, or meiosis, if we're talking about a reproductive cell that's dividing, like an egg cell or a sperm cell. Remember too that cytokinesis is the physical process that pinches off and splits the parent cell into two daughter cells. That is a visible thing occurring inside an animal cell. It's harder to see cytokinesis in a plant cell because instead of having a pinching off event, instead new cell wall will be laid down. And that one's not quite as easy to see. Now, there are certain regulatory checkpoints inside the cell cycle that help ensure that all of this copying and replicating that's going on is done correctly. Because as you can imagine, it's critical that each daughter cell have all of the things that it needs in order to be a healthy functioning cell. And of course, you could argue that the most important thing that the daughter cell needs is an accurate copy of the parent cell DNA. So the checkpoints that we talked about during the cell cycle material include checkpoints at the end of the gap phases, the G1 and the G2, and a checkpoint that's smack in the middle of the M phase. And these are the things that are going to ensure that the parent cell has been carefully and correctly copied in every aspect before one copy of each set of material is passed on to the daughters. So the checkpoints at the end of G1, or the checkpoint at the end of G1, ensures that the cell is ready to go into S. And S, of course, is where the DNA will be synthesized or replicated. The checkpoint at the end of G2 makes sure that the cell is ready to go into M, which is the time when the nucleus is going to divide. Every effort that the cell can put in is made to ensure that the DNA that's being passed on to these daughter cells is an accurate and full copy of the parent cell DNA. So, today we're going to specifically be talking about mitosis, the M phase. And remember, M doesn't always stand for mitosis. M only stands for mitosis when we're talking about a somatic cell, a body cell, like a skin cell or a liver cell or a kidney cell or a blood cell. We're going to talk about meiosis in a couple of weeks, and meiosis is a similar process of nuclear division, but it happens in a reproductive cell, like a sperm or an egg cell. So, the M phase, and today that's going to stand for mitosis, is the time when the replicated chromosomes, the copied chromosomes that were created during S phase, are going to be divided up so that one full set can be passed into each daughter cell. So if we're going to define mitosis, we would say that it's a form of nuclear division. It's a form of nuclear division that results in the formation of two brand new daughter nuclei that are genetically identical to the parent nucleus. So this is a process that's going to occur in eukaryotic cells, because only eukaryotic cells have a nucleus, and it's going to occur in somatic cells. If you're hearing any barking in the background, that's due to my dogs playing while I'm recording this lecture. I don't know if you can see them, but let's see if I can get them on camera. So we've got the old fella, Cooper, and we've got the two young guys. We've got the puppy, Bonnie, and the older girl, Winnie, and right now they're biting at each other's face. Sorry about that. Okay, let's take a look at this slide. This is a picture that comes out of your textbook, and I like it because it tells us when, in the life of an organism, mitosis and meiosis are likely to occur. So, in this slide, you can see a dog on the left-hand side, a German Shepherd, and what they're showing us is that this German Shepherd developed from a fertilized egg. So that was the first moment that this German Shepherd was an organism. And through all of the growth of that fertilized egg, through the embryo stage and through the fetus stage, and when that puppy was born, and when that puppy grew into an adult dog, all of the cell division that made that happen was mitosis. So this is the kind of cell division that's responsible for growth and development. So all mitosis occurred in order to produce an adult dog. Thank you very much for joining in, Cooper. Now, you'll see that throughout the life of the adult dog, mitosis is also occurring. And this is the time when the cells are being replaced and repaired and so on. The only time we see these blue arrows, these blue bars, are the times associated with meiosis, and those surround the production of reproductive cells. So meiosis occurs to help us make the egg cell and the sperm cell, or the gametes, and it also happens shortly after the production of those cells, up to and including the time of fertilization. So you can see the time where an egg cell and a sperm cell come together and create a fertilized egg is actually pretty complicated, because there's some meiosis still going on to help produce that brand new fertilized egg, that brand new organism. Now, in this video, I want you to take a look at the material in the center of the cell. This is actually chromosome material that's inside a nucleus, inside a cell. So all of these sort of darkened, squiggly lines in here are condensed chromosomes. Remember, you can only see a chromosome when it condenses, at least underneath a light microscope. So if I set this into motion, what you're going to see is these chromosomes start moving.
Taken from a condense.
These chromosomes start to move, and you start to see the activities of mitosis.
Next, the mitotic spindle lines them up in the center of the cell. At the metaphase to anaphase transition, the sister chromatids of every chromosome pair separate suddenly in striking synchrony. The chromosomes are pulled along the microtubules of the spindle to opposite ends of the cell. After chromosome separation, membrane vesicles line up in the center and fuse with each other to form the new plasma membranes that separate the two daughter cells. At telophase, the chromosomes decondense in the newly formed nuclei.
Now we'll take a look at that video again at the end of this material to see if we can follow through stage to stage to stage. So, during the synthesis phase of the cell cycle, the DNA is being copied or replicated. And this is the process that we call S, the S phase of the cell cycle. During mitosis, one set of those chromosomes will be distributed into one daughter cell, and one set will be distributed into another daughter cell, so that each daughter cell gets her own full set of chromosomes. We can break mitosis into 5 distinct phases. The first one's called prophase, the second one's called prometaphase, then we have metaphase, anaphase, and telophase. And of course, at the end of telophase, in an animal cell, we would have cytokinesis, that actual pinching off of the cell membrane. Now you'll notice I have prometaphase here in parentheses, and the reason I do that is because prometaphase is a very short stage in mitosis, and it's very hard to see. We'll take a look under the microscope in lab this week at all of these stages, but don't be surprised if we don't get to see prometaphase. It's often missed. So the first stage is called prophase, and literally that translates as the before phase. In some material you'll read this phase is called early prophase, but we're going to call it prophase. Okay? And it's at this time that the chromosomes first condense and become visible underneath the light microscope. In the cytoplasm, a structure called the mitotic spindle is going to form. Now, this is an array of microtubules, sort of like building a scaffolding that's going to help us physically pull the 2 sets of chromosomes apart. It provides the structure to do this and also the mechanical force to do this. So when we talk about the mitotic spindle, we have to talk about a little bit of new vocabulary. It turns out that in a cell, the mitotic spindle grows out of a structure called the centrosome. So the centrosome is a structure, an area, if you will, inside the cytoplasm that contains a pair of what we call centrioles. Centrioles are also called microtubule organizing centers. So this is the place where the scaffolding of microtubules first starts to grow. And as they grow, they create what we call spindle fibers. Something important to remember is that spindle fibers are not ordinarily visible underneath a light microscope because you have to stain for them very specially. So we forget that the mitotic spindle is actually in place helping pull these chromosomes apart during mitosis. It's important, though, to remember that the spindle is critical for this process to occur. So inside this slide, you can see just a very generic diagram of an animal cell with a nucleus and some condensing chromosomes. And outside in the cytoplasm, we can see this structure starting to appear. Right over here, we have a centrosome, and it contains a pair of centrioles. Notice that centrioles always occur in pairs, and they always sit at a 90-degree angle to one another, so that when the spindle fibers start to grow out of these centrioles, they grow out in all directions and help to start to build this mitotic spindle. Here's one centrosome with a pair of centrioles, and here's another centrosome with a pair of centrioles. And here's the beginnings of the mitotic spindle. This is prophase. In your book, prophase looks very similar, but remember, in your book, you only have 4 chromosomes in your individual. 2 long chromosomes and 2 short chromosomes. So when we look into the nucleus at mitosis, since we've gone through S, or synthesis, we now have 2 copies of this one, and 2 of this one, and 2 of this one, and 2 of this one. So we now have 8 chromosomes. Over here, we have the beginnings of the mitotic spindle. We have a centriole inside, we have a pair of centrioles inside a centrosome. We have another pair of centrioles inside a centrosome. And we have these early mitotic spindle fibers. So another term that you can see on this slide comes up when we start talking about our next stage, which is prometaphase. In prometaphase, our duplicated chromosomes, our copied chromosomes, are going to connect with one another. This connection point is called a centromere, And inside the centromere, there's another structure called a kinetochore. At prophase, two important things are happening. First, the nuclear envelope is breaking down. It's disappearing and it's releasing the paired copied chromosomes into the cytoplasm. In addition, the spindle apparatus is moving into position. So now we have a pair of centrioles at one pole in the cell, the north pole if you will, and we have a second pair at the south pole, and our spindle fibers are stretched all across the cytoplasmic area of the cell. One spindle fiber from each centriole pair is going to connect with each pair of chromosomes at the kinetochore. So in this slide, we have this yellow spindle fiber coming in and connecting with that little black dot on the top, and down here we have another spindle fiber coming in and connecting with that little black dot. Okay, so nuclear envelope breaks down and the spindle fibers move into place and connect at each kinetochore. So what does this look like when we actually look at a living cell? Because it's not going to look as pretty and complete as it does inside your textbook. Let's take a look at what we're going to be seeing in lab especially. This is a preparation of plant cells, and I know that because these cells are squared off due to the presence of a cell wall. So we've got a cell here, and we've got a cell here, and here, and here, all the way across the field on the microscope slide. Now, most of these cells have this nice sort of homogeneous red-pink circle in the center. This is the nucleus. There's a nuclear envelope around this material, and there's this homogeneous looking chromatin inside. These cells, the cells that look very homogeneous, just a nice round pink red area, these are cells that are in interphase. These are cells that are either in the G1 stage, the S stage, or the G2 stage. This is interphase. We can't tell which stage they're in because we can't see the events occurring until the cell goes into M. And what you're seeing in this nucleus and in this nucleus is just that. These are two nuclei that suddenly look very different. Suddenly there's all these sort of spots and lines in here. Those are chromosomes condensing. So we are now in prophase, or what's on the slide is early prophase. The other thing I want to point out is right here. See this little darkened area in here? We have two darkened areas in this nuclei. And if you look very carefully, even in the interphase nuclei, some of the cells have these darkened areas. These are called nucleoli. The singular of the word is nucleolus. This is a darkened area inside the nucleus, and it's a place where RNA is being actively synthesized. So a place where transcription is occurring at that moment. So this is a nucleus that's in prophase. Here's its nucleolus. This is a nucleus that's in prophase, and it has two nucleoli. The chromosomes are condensing. The spindle apparatus is forming out here. We just can't see it. And the nuclear envelope is still in place. When we go to prometaphase, remember, a couple of things are going to change. The nuclear envelope is going to break down, and the chromosomes will be released into the cytoplasm. We'll still see them, but they'll look very unorganized. The chromosome pairs will be attached to each other at a structure called the centromere, and we're going to give these chromosome pairs a name now. We're going to call these sister chromatids. Sisters because they're identical to one another, and chromatids because they are condensed chromosome material. At the prometaphase, the spindle fibers will be in place and will attach to each pair of sister chromatids at the kinetochore. Remember, this is not a stage that we typically are able to see.